Okay, guys, here we have some more. All right, let me begin. As you all know, my name is David Arieti, and I've been teaching here for over 30 years. And every year we celebrate Earth Day. And so I like to raise a little hell on Earth Day because my main field is the environment. So I figure I might as well talk about it. And my other field is oceanography and the organisms that live in it, especially the microbes, especially little things called algae. So today we're going to learn about one specific type and about the problem it has caused in both fresh water and possibly now to salt water. So let's begin. So the name of this lecture is called the Hysteria of Pfiesteria, and it's a quite interesting organism. And the word Pfiesteria comes from a woman scientist, a marine biologist named Neuse River. And the species name, by the way, is called Pfiesteria, which means fish killer. And this one has killed over 1 billion fish. That's right, with a B. And we're going to learn about this organism and what's happening with organisms related to it. So what I want to do is first tell you about the type of organism that this is, and then I'll go into this one. This one belongs to a group of organisms called dinoflagellates. And let me tell you how that fits in to the picture of all life. We divide life up into 6 kingdoms: the fungi, the protists, the plants, the bacteria, the archaebacteria. They differ from other organisms because of their cell wall, other bacteria, because of their cell wall, and the animal kingdoms, okay? Now, I should point out that many people have called me a fungus because they say, gee, Dave, you're a real fun guy. Just thought I'd mention it. Okay, now, we have lots of types of algae, okay? These are the main groups of algae that exist on the planet today, and I'm going to show you them. But the one we're mainly dealing with is this middle one called the Pyrophyta or Pyrophycophyta, which means fire algae. The organism I'm talking today is about that, but I just want to show you how all of these fit in. And by the way, all these belong to the kingdom Protist, at least for now, because classification is always changing. And at least for now, though, we call them a Protist, because when I went to high school, which was in the 50s, we only had 2 kingdoms, animals and plants. So they would put the little amoebas into the animal kingdom and the bacteria and the fungi in the plant kingdom, but then they realized there's more to it than that, so they decided to put all those in both the fungi and the protists, and the protist, the group, as well as the bacteria. Whoops. Okay, hold on a second. What's going on? Here we go. Let me get this out. I pressed the wrong button. Okay, yeah, that happens. Okay, these are the pictures of the types of algae that I just mentioned. The Euglenophyta, the Chrysophyta, which are diatoms, they're very pretty. There's the Xanthophyta, which are yellow-green algae. The Chlorophyta, which are green, which are plain green algae. I notice you see a little orange there, but that does contain some other pigments. Then we have the red algae, the brown algae, the cyanobacteria, which are actually bacteria, but we call them the blue-green algae because they're interesting and they behave like algae. But even though they're bacteria, we include this specific type. And as you see, there's some water. And the trouble with these is these also produce a toxin which can really make people sick and kill them. And there was some outbreaks of these which took place in Ohio a number of years ago. It produced a substance called microcystin, and that caused a lot of problems to the people who lived in Ohio and had to drink the water. But today, we're actually going to talk about the group that includes these organisms. These are the dinoflagellates, or the Pyrophycophytas, which literally means fire algae plant. The common name is dinoflagellates. Why dinoflagellates? Dino means whirling. And they whirl because they have 2 types of flagellum. A flagellum is a long whip-like structure, and you can see it here using this scanning electron microscope. That's this one there. These are all light micrographs except for this one. But this one and that one were taken with a scanning electron microscope, and my class knows about that because we spend a lot of time discussing images from microscopes. The rest were taken with the light microscope, and you can't see as well with the light microscope, but you can certainly see the clarity with the scanning. Many of these cause the red tides, and we just had one a few months ago off the coast of Florida, and by the dinoflagellates. And so that's what they look like, hence the term red tide, because these things multiply like crazy, and many of them have toxins, and we're going to talk about those in a few minutes. Now, I want to give you a slight introduction to the dinoflagellates, and I'm going to play a little movie for you. It lasts about a minute, somewhere in that neighborhood, and I go through all these efforts. Let's hope the sound works. And let's play it.
Dinoflagellates, dinophyta, are mostly unicellular organisms, although a few form colonies.
Here we go.
Their cells are often covered by shells composed of interlocking cellulose plates. Dinoflagellates are equipped with two flagella that set the cells spinning like a top. Most dinoflagellates are photosynthesizing organisms and contain chlorophyll A, chlorophyll B, and carotenoids. Polysaccharides are the usual storage materials found in dinoflagellates. Dinoflagellates are mainly marine organisms, although there are a few freshwater species. Most of them float freely in the body of the water. Dinoflagellates are one of the major components of plankton in warm, temperate seas. A few are parasites of other protists and fish. Others are symbionts of anthozoans and medusas.
Okay, that's good. At least we got that working. Now, I want to continue. Let me just close the tabs here. Okay, so that gives you somewhat. Now I want to show you what they look like when they swim. And these are all light. This was taken through a light microscope. It's important that you see how these things move so you can see why they're called whirling. And where are you there? There it is. Come on, up, 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 and it's cooked. It's getting there. There it is. Okay, then we'll go here. Let's make it bigger. There we go. There it is. See it? That's the way they move, okay? There, you see it? That's that one. And the black thing in there could be an eye spot or the nucleus. It's hard to tell. Let's see, there it is. You see it? Now that's a typical dinoflagellate. They move quite quickly and they have a whirling motion because of their flagellum. And I'll just show you this one. All of them move in a similar pattern. And these, by the way, are responsible for much of the photosynthetic activity in the oceans, okay? Those and something else called the diatoms. But I'm not going to speak about the diatoms today, maybe a different day. Over here, we have a good anatomical picture of a dinoflagellate, again, taken with the scanning electron microscope. If you look here, you'll see one flagellum here. This is a trailing flagellum. We also have a lateral flagellum and transverse flagellum. That's located in something called the cingulum, and the sulcus is where the trailing flagellum is. These are very good pictures. With all this technology, I like to make use of everything we have, especially technologically wise. Now, I have here a list of the certain characteristics of dinoflagellates in general. These are real unique organisms in that they have many characteristics which are only found in this type of organism. Now, I don't want to go into all of these, but I'll just mention them briefly. These are all eukaryotic cells. Everybody in this room is composed of eukaryotic cells. Essentially, that means we have our cells contain a nucleus and they have a membrane surrounding the nucleus, whereas things like bacteria do not have a nucleus. They all have only one chromosome. So bacteria have one chromosome and the eukaryotes have more than one. Humans have 46 chromosomes. Chimpanzees have 48. There's a flower or a plant called adder's tongue fern has 1,262 chromosomes. As far as we know, that's the largest number of any organism. And again, that's a plant. Anyway, they all have different types of chlorophyll. There's around 2,000 known species of these. Many contain a covering called a theca. The ones with a theca are called thecate. That's an outer layer like an armor. The ones without the armor are called athecate. And the theca also has a strange name called amphiesma. Many species are heterotrophic as well as autotrophic. What does that mean? That means that those species which are heterotrophic eat, like you and I do. They actually gobble up other organisms. But some of them, in addition to eating, can also photosynthesize. So that means they take the light from the sun and make their own food that way. So that's pretty cool. And you have both freshwater and salt species. But the most interesting part about all these dinoflagellates is they have a special nucleus called a dinocaryon. Dino means dinoflagellate, and the suffix caryon means nucleus. This is by far one of the most interesting types of organisms ever. And anyway, we do have some more facts here. I'll go into those in a minute. But many of these also cause disease, and some are bioluminescent, which I'll get to. And they contain other things which I don't want to cover here. And I'm going to show you the nucleus in a few minutes. Now, many of these are toxic. There used to be only 30 years ago, they discovered only 22 species were toxic. Now, over 60 species are toxic. So what's causing this problem? Well, the problem is that they are mutating. Now, why do they mutate? I don't really know, but something's going on there in the environment. It could be either the climate or other things which are causing these to mutate, because the mutated ones are quite toxic and they're causing a problem. And this could be something why they're mutating. We have something here called endosymbiosis, which means they gobble up another organism and then it stays with them and they keep multiplying with that other organism, and there's some interesting phenomena associated with this topic called endosymbiosis, where if you look at each one individually, they're gobbling up another type of organism, and that organism becomes part of the dinoflagellate. So it's quite interesting how this goes on. And by the way, these two pictures of two dinoflagellates, I am proud to say, I took these myself in 1970 at my first attempt at doing microphotography. These come from Israel. I was doing work on the Sea of Galilee, and this is the ones I took. Can you imagine? This was done about 1970. So based on my calculation, that seems to be about, what, 49 years ago? So these are one of the very first photos I've ever taken myself, so I'm proud of them. And this one, by the way, is called the Serratium hirundinella, which is quite common in the freshwater. These are both freshwater. This one's known as a peridinium, and they like to live together, these two together. You always find them, well, not always, but in this particular lake, the Sea of Galilee, that was the lake where Jesus walked on the water. I did work there for 2.5 years, and so I was lucky enough to work there, and we had microscopes and cameras and everything, so I was able to take advantage. And by the way, here's an organism called, now I didn't make this up, called Serratium arietinum. It has my name. And so I put my picture there to see if there's any resemblance. I don't know. But really, this is the real name. And my last name is spelled Arieti. And I happened to find this, so I thought that's cool. And if you go online, you'll see Serratium arietinum. So I figure, I don't know, see if there's a slight resemblance. I'm still looking for it, but that's the real picture. What can I say? If anybody notices a resemblance, let me know. All right. Now, here are examples of a thecate and athecate one. In other words, an athecate one is also called a naked one because this one does not have a special capsule, where the other one does, okay? And again, the capsule is called a theca or amphiesma. Now, here is the picture of the nucleus I want to show you. Those of you in my class know that when we talk about the nuclei and the chromosomes look like this. there's a long strand of DNA, which we all hear about DNA, right? We have 23andMe. Remember these kits, you want to find out your DNA, so that's what they do essentially. They take the DNA. Our DNA, it's like a spool of thread. Inside the cell, it has to stay there. How does it stay there? Well, it has to be wrapped around something. It can't just be messy. So in a normal cell, they're wrapped around little proteins, 8 of them, for each 2.5 turns, and those are called histones, not to be confused with hearthstones. Just thought I'd mention that. All right, anyway, have fun with the English language, why not, yeah? All right, anyway, so it's like a spool of thread. Now, the dinoflagellates, they do not have that. So they're in a constant state of packaging, you might say. You see? This looks different than a normal nucleus. So this was taken with a light microscope. You see it? Okay? So as you can see, here, I'll go over here, show you that too. Over here, these, over there, that's a dinocaryon. That's different than the other types of cells. Hey, you know what we can do is shut off that light. Maybe we can see better since we have the light outside. We got to take advantage of solar energy. By the way, everybody in this room is a result of solar energy because everything you ate came from the sun, right? Even if you eat the cow eats the grass, right? By the way, speaking of cows, you know why cows have bells? Because their horns don't work, which is what they make in patients. All right, anyway, now, so if we look here, now this is a regular light micrograph. This other one, this black and white, is a transmission electron micrograph. And as you can see, it looks, these are the chromosomes, and you see they're condensed. In other words, they're coming together. Okay, all the time, the chromosomes exist this way. So because of this, the scientists call this a dinocaryon. Again, the suffix "cary" means nucleus, and dino comes from the word dinoflagellate. And by the way, the word dino could have 2 meanings. One means whirling, and the other one means terrifying. Now, I didn't make this up. That's what it means. So you might find that in the literature. Now, I wanted to compare the dinoflagellate nucleus with a normal nucleus of an organism. So here we have a paramecium splitting in 2, and we have a green algae called a chlorella. Okay? And by the way, the word paramecium reminds me of something because I always ask somebody, what do you call 2 meciums walking down the street? The answer is a paramecium. I just thought I'd mention it, a little extra there. All right. Now, in addition to what I just told you about the dinoflagellates, many of them have symbiotic relationships. This one in particular makes up something called the zooxanthellae, which is another name for this dinoflagellate, which occupies coral. Okay, corals, they depend. They're related to the jellyfish. Okay, they belong to the Cnidaria. However, the corals need light to survive. That's why most of them are near the surface. So they have algae species living inside the corals. And these corals get their energy and food from the algae. And we're worried about this now because of climate change, and that's causing a lot of bleaching of the corals. And that's very important because the coral reefs are the basic, it's like the Grand Central Station in the ocean. That's where most of the fish spawn there, you see. So this is a big issue that we have to worry about amongst other things. Anyway, so a dinoflagellate species occupies the coral. Now, here we see again a scanning electron micrograph of the corals, and where you see the term symbiodinium, those are the actual algae stuck inside the tissues of the corals. Now you notice I spend a lot of time on the electron microscope here and the other one because we're basically talking about microorganisms, and we can see them a lot clearer. Now, I want to show you a couple more things before I get on to the Pfiesteria itself, because I want to give you an introduction to the dinoflagellates in general. They also practice bioluminescence, which means they glow in the dark. Now, in the world there are 5 bays specifically where we have bioluminescence of these organisms. And here they are. There's one in Jamaica, in Vietnam, in Puerto Rico, Mosquito Bay, and I'm not sure where that is, but there's about 5 of them in the world. Now I'm going to show you a quickie movie on bioluminescence, and here you see fish swimming in the water, and as they swim, the water glows. It's pretty cool. But so far, everything's worked. There it is. There it is. He plays music, maybe he narrates in Spanish.
Mar de Cortés, México.
Yeah.
Por la razón que sea la magia creada por la...
I'll shut off the sound and I'll just play it for you. Whoops, there it is. Up here. Oops, wrong line. I gotta go back. Sorry. There we go. I'll just leave it.
Mar de Cortés.
See the blue? All that's not light. Oh, that's not light from the photographer. That's light from the ocean as it moves. It causes the water to glow because its enzymes are being produced from the dinoflagellates, which make them glow. See the fish? Isn't that cool? This is not this is not
Delfines a la cadera se revelan como resplandecientes siluetas.
It's actual bioluminescence. So this is what they do with the dinoflagellates. They're real interesting.
Siempre que
I
aparece
want to point out too that many of the luminescent ones are not toxic, but it's mainly the
Es posible que
photosynthetic
los
ones
delfines
which
se
are
orienten hacia
toxic.
sus presas por la luz de los dinoflagelados.
Isn't that cool? For those of you who practice your Spanish here, listening.
Hasta ahora no había sido posible grabar estas escenas con tanta claridad. Pero cada noche se producen espectaculares juegos de luces como este en alguna parte de la inmensidad del océano.
Pretty beautiful. Okay, anyway, you get the idea. Now, I have here a picture of them on, look at that, on the coast. So, this group of organisms, I'm sure many of you have never heard of it. You did hear of the red tides. So you see some of the interesting facts about this particular organism. Now, how do they identify the thecate ones, the ones with a theca? Well, the scientific formula depending on the capsules of these things in the theca. You see, there's like a special code that they use to describe it, okay, of all the hundreds known that have a special type of theca. Now, what I want to do before I get to the Pfiesteria is talk to you about the diseases caused by dinoflagellates. And here you see the major ones, the ciguatara poisoning, paralytic shellfish poisoning. That's a quite interesting one. Let me tell you about that real quick. How would you like to go to a picnic on the ocean and you eat some clams, okay? It doesn't matter whether they're cooked or uncooked. They contain a poison which is not heat labile. That means that the heat does not destroy the toxin. So you eat these, and then a half hour later you begin to feel a tingling sensation in your mouth, and then you have trouble breathing, and within an hour or two you might be dead. This happens because when they take the shellfish from areas they don't know, and they came from an area which had a red tide, you can get that from that way as well. Also, you have neurotoxic shellfish poisoning, which hits the nerves, diuretic shellfish poisoning, as well as another one which I didn't put here, one called domoic acid shellfish poisoning, which causes amnesia. So they call it amnesic shellfish poisoning. So you got to watch what you eat. It's not a good idea these days to eat shellfish of any kind because of that. And also, we're going to be talking about today the major one, Pfiesteria, which is the name of the poisoning that you get from even being around this specific toxic microorganism. Okay, and by the way, these are the species which cause the diseases that I just mentioned. These are all different types of dinoflagellates. Again, the top two are the electron microscope. The other ones, with the exception of the lower right, are light micrographs, and then the bottom one, of course, is a sort of a conglomerate of a scanning electron microscopes. Now, in the 90s, a fellow named Rodney Barker wrote this book, and the waters turned to blood. Sounds like a biblical book, but it's actually about the problem of Pfiesteria piscicida, which is what I'm going to be talking about. Pfiesteria, I know it rhymes with hysteria, but it came from the name of a woman scientist named Lois Pfiester. And Rodney Barker wrote a wonderful book about this. And this cell has a nickname. It's called the cell from hell, okay? So that tells you how lovely it is. Okay. Now, I want to give you some information about Pfiesteria. And I know it spells kind of strange, but that's the way you spell it. And it's a toxic dinoflagellate associated with fish lesions and fish kills in the coastal waters from Delaware to North Carolina. And I'm going to tell you some little political stuff involved with this too, which makes it frustrating that the health department, especially of North Carolina, didn't want to recognize there's a problem. And I'll discuss that in a few minutes. Now, as a rule, this organism has both photosynthetic and non-photosynthetic components. It was discovered in 1988, and there are three typical forms: amoeboid, flagellated, and encysted form. An encysted form means that it protects itself. The nucleus and the whole body of it is surrounded by a hard capsule, and it prevents it from being destroyed by various environmental conditions. When environmental conditions are right, they'll come out and germinate. Germinate, by the way, does not mean move to Germany. It means to grow and cause problems. Now, there's also something very interesting. This dinoflagellate has 24 life shapes, life forms that they discovered. You see, that's why this is so fascinating. 24 types. Depending on the type determines how toxic it is, okay? And by the way, they did find some other types of Pfiesterias or Pfiesteria-like organisms, and I'll mention those also. Now, as for toxicity of these things, they figure 150 cells per milliliter is enough to kill some fish. Yet they found concentrations over 600 cells per milliliter. Now, over here, I have a bottle with water and a blue dye. This is exactly 1 liter of water. I dyed it blue just to show you so you can see it easy. I don't know if you can see it here. But in theory, then, if this were 1 liter of water, this would have 150,000 cells, which is enough to kill a lot of fish. You see? Just what's in this little container. That's why I have it. And by the way, I also brought the book here too, if anyone wants to see it. Here it is, and the waters turn to blood. Very interesting book. All right, so there's 1 liter. 1 milliliter is equal to 1/1000th of a liter. Okay. Now, how does this affect fish? Well, it's real interesting. Again, this is like a science fiction movie. What happens is, you have a fish in the water. They have various excretions. The Pfiesteria rush to the fish and start causing a problem, okay? They like rush to it. It's like a pheromone. A pheromone is a substance produced by an organism to either attract or repel other organisms. And this obviously, whatever these fish excrete, the fish zap to it, come to it immediately. It's like a call, hey, come to me guys, come to me. That's what the excretions tell the fish. Now this is what one of these organisms look like. And again, I didn't make this up. It's called the cell from hell. Looked kind of interesting. This one actually, you might say, looks like it has 2 eyes and a nose, doesn't it? That's just another. Here, I'll show you over here on this group here. Looks like, again, this is a scanning electron micrograph. See the 2 eyes there and the nose? Pretty interesting looking organism. And anyway, that's what it looks like. Let me show you. Well, I didn't want to do this. Let me show you this too. Just to show you how to make that cell from hell stuff up. This thing comes up and we should get, there it is. See? This is by a person named Booz Bayathavam. Anyway, so there's a lot of information, and they do call this nickname the cell from hell. I just want to let you know I didn't make it up. And I know it was in the book too. All right. Now this woman, she's actually the heroine of all this. Her name is Joanne Burkholder. She actually discovered the organism in the Neuse River in North Carolina. Now here's the problem. She wanted to convince the health department. There was a fellow named Ron Levine. He belonged to the DEHNR, which stands for Department of Environmental Health and Natural Resources of North Carolina. The people didn't want to know that there was a problem in their water, something like the movie Jaws. I don't know if you saw the original Jaws, when Murray Hamilton, he played the mayor of the town where that happened. He said, you got to keep this quiet. You know, we're going to lose money if people stop coming here. You know, I remember that scene in the movie, and that's exactly what happened here in North Carolina. North Carolina has a big fishing industry and tourist industry. So if there's something here preventing people from going swimming and fishing, you know, there goes your tourist business. Put another way, there goes your billions of dollars, you see? So here, in the long run, everything refers to money. So anyway, I just want to mention that. So this book basically tells the story of Joanne Burkholder and her assistant named Howard Glasgow, the trouble they had in convincing the government that there's a real problem there. And yet, again, all they cared about, the people in the government, was the money that would be lost to North Carolina. And by the way, it still persists, I think, a little bit. We still have that problem, and it's spreading. I think they found this organism in Europe now. Anyway, so in my book called The Earth is My Patient, and I do have a few copies here if anyone wants to buy one. We have the special extortion price of 2 for the price of 10, or we'll make a deal with you. Yeah, you know. Anyway, I wrote about this particular organism here, and let me show you what I wrote. Now remember, I said the Neuse River. So, in a section on the Pfiesteria, this comes from my book, I have the section, no noose is good news. Yes, noose is bad news. You get the idea? Because noose sounds like news, you see? See the point? So actually, this is from my book, and it's in the back of the book near there. And if you read the first part, it says, yes, reader, you read the above correctly. The Neuse River is a river in North Carolina. Its existence was not widely known, at least by me, or to much of the world's population prior to 1996. It was known to those who lived near it, or to those who were interested in doing research on North Carolina's river. Since then, it has become famous, or should I say infamous, because of a microscopic resident nicknamed the Cell from Hell. This sounds like an introduction to the Twilight Zone. Anyway, I just wrote that, and you get the point. Now, what kind of symptoms does this have? This is good for those of you who are in the medical profession or who want to be in the medical profession. These are the symptoms, and I'm going to tell you some stories specifically about what happened to a few of the actual workers, the scientists who work with this. You wouldn't believe what I'm going to tell you, but it's true. They have a whole bunch of symptoms: respiratory problems, fatigue, eye irritation, headaches, skin rashes, muscle cramping, gastroenteritis, nausea, stomach cramps, vomiting, cognition. Do they recognize people? Personality changes, and most of all, memory disturbances. So now what I'm going to do is relate some stories to you about this. Now this I got from the book. I want to tell you about certain people that were exposed to this. Now, remember, these are workers working with this organism. At the time they were working with it, they didn't realize how toxic it was, okay? Let me tell you, I know for a fact that if you go on a boat over the red tide, let's say a red tide, with different type of organisms, you get sick just by breathing the air because you have the ocean spray, you see? So these red tides, when you have such a high concentration of these organisms, they cause a severe problem, especially, you know, if you want to breathe it in. And it makes you nauseous, makes you sick, and things like this. So, let me read you this about one of the workers, a scientist, who actually worked with these organisms in a tremendous quantity. And I'm going to show you now what they decided, how to protect yourself. If you want to work with these things, you have to protect yourself, and I'll show you what type of systems we use. Okay, but first, let me show you this. This took place before they actually used protective equipment, such as a special mask, air being pumped in, and all this. The memory dysfunction was an inability to create new memories, similar to Alzheimer's, like suppression of short-term memory and learning disabilities. For days or weeks following exposure, several laboratory personnel could recognize words individually, but could not form sentences, perform simple arithmetic, or remember more than the last words of a sentence directed to them. Can you imagine? These were scientists who worked with this, and then they forget how to do simple things that you and I, that even a third grader can take for granted. Look, the most seriously affected person in our laboratory, who is a highly intelligent researcher, managed only a 7-year-old's reading level for 3 months after exposure, and required reading lessons at first to regain reading ability. The person recovered 3 months after exposure. What do you make of that? Here you have PhDs, and they forget how to read, how to form sentences. Incredible. Now, if you look at this picture, this is what they do to the fish. You see? Now, I didn't make this up, but wait, it gets worse. Here we see more fish, always around that spot near the tail region. Here we see a whole bunch of these fish. But wait, how'd you like that? Okay, it's a fish kill with thousands of fish. You know, it's amazing to me, every time I see a fish kill, I saw one of the herring, I think it was down in the Indiana part of Lake Michigan one day. I went there. I saw a whole bunch of dead herring, and they said that was due to a natural virus that happens every year. But these were caused by Pfiesteria, you see. So, when you had these fish kills, the people along the Neuse River were panicking. They wanted to know what's going on. And of course, it's obvious if you have a fish kill, there's some problem. At first, again, I have to use the word lie, because the Department of Health and Natural Resources said that it was low oxygen content. Actually, they tested the oxygen. That was okay, you see. Fish, by the way, need at least 5 parts per million oxygen to survive, okay? I think humans need at least 19%, okay? If you live in an atmosphere, you need 19% oxygen. At the moment, in this room, we have about 20.94%, okay? That's the average oxygen we have on the planet, but anything below 19% might cause a problem. So if we read some of this information about the blooms, we say here, Hysteria may produce toxins that numb fish, allowing the microbes to feed on the fish. High concentrations of Pfiesteria can cause deep lesions, as you have seen just now. Blooms, which is a tremendous amount, usually occur, and they might last for a few hours. Massive fish kills in the estuaries along North Carolina occurred. Now, as I mentioned earlier, these things are so strange that there are 24 different shapes which have been identified. That's what makes them unique. Here are some of the forms. The one on the right is probably the most hazardous. The colored picture taken with the regular microscope, a light microscope, that's called the Lobos amoeba. And the picture next to it, here, that's the scanning one, right? Whoops, what happened? Uh-oh, there we go. I keep messing with this. Oops, let me get this. These things here. Modern technology is good when it works, you know. Yeah. Okay, anyway, let me just, I want to put the, there we go. That one there, and this is called a Philos amoeba because it has sharp needle-like projections, or pseudopods. Philo means long, wiry-like. That's the most toxic stage. And by the way, this reminded me of the movie Life. I don't know if you saw the movie Life. That was the science fiction. In the space station, they get, I think, an asteroid, and it contains one of these creatures. And anyway, at the end of the movie, you get the impression, I don't want to give it away, but I'll give it away anyway, the whole world goes kapooey because they went back to Earth, and anyway, you get the idea. I don't want to give more away. Quite an interesting movie. I enjoyed it. Anyway, here we see some more effects of this. And here we see some more different shapes. And this one's quite interesting. This one, we have 2 basic shapes of the amoeboid type. And this is eating something called a cryptomonas. A cryptomonas is another algae. Actually, it's a monad. Monads are rare compared to the other species of algae. But if you see it there, it's that little green thing being engulfed by this, that's a Lobos amoeba. And the bottom one is the Philos amoeba form of Pfiesteria. Okay? So this one actually likes to eat a whole bunch of stuff. Here we see some more This is the toxic stage. A zoospore is actually a spore that has a protective coating and has a flagellum that makes it swim. Now, how do you examine certain types of microbes? I want to explain this to you. This is important that you understand. They've labeled these biosafety levels 1 through 4. So BSL stands for biosafety level, okay? A biosafety 1 looks like this. Essentially, all you need is a lab coat, not much. We teach here at this campus microbiology up in the biology lab, okay? All the students just have a lab coat, okay? They have a white lab coat, that's all you need, because they're not really dealing with infectious agents. You know, we can deal with non-pathogenic strains of various organisms, such as E. coli and some other things, which are quite common and which do not cause any problems. A level 2 biosafety area, you work with slightly more hazardous organisms, okay? Equine encephalitis virus, they cause the horse problem in horses. And then we have Staphylococcus aureus, staph infections. These are not incredibly poisonous or dangerous yet. They use, that's biosafety level 2, where they use a hood. And you know, it's like a big box and has a fan and a window in there. It's like where you do your work inside the box, and notice you wear gloves. A level 3 biosafety zone includes work on microbes which could be quite infectious, including yellow fever, Niles West virus, tuberculosis. By the way, tuberculosis is caused by the same genus of organisms as leprosy is. And then, of course, a level 3 Pfiesteria. Notice the way that guy is dressed. He has a mask on and he has pumped air in, and it's known as positive pressure because you blow the air in the mask. In other words, the air is going in. We use that term with positive pressure. So air can go out, but nothing can go in. You understand? So if you have a small leak, it'll blow out whatever substance you have. And then the level 4, which is the most stringent, looks like they're spacesuits, and actually they are. A level 4 is an area where, in order to enter, first you go into a room, you change your clothes, you take them off, and you put on these spacesuits. They're like triple layer. You have triple layer gloves. You have to have air pumped in, just like a spacesuit, a suit. Then you go to an airlock, okay? It's just like leaving the space shuttle or something to go outside. You go into the airlock, you close the back door, and then you open up the regular door where you go to the lab, and you have to work. Can you imagine doing microscopy in a suit like that? This is for real incredibly toxic microbes such as the Ebola and Marburg virus, okay? Now, for those of you who saw the movie Outbreak with Dustin Hoffman, that movie was essentially based on The Hot Zone written by Richard Preston. And the Hot Zone is this level 4 biosafety facility. And I suggest if you want to read more about the Hot Zone, read the book. It's about the Ebola virus and how it started. And the Hot Zone is actually, again, the BS level 4 area of the lab, which was Usamaride. By the way, we're going to mention Usamaride here too, which really disturbed me, and I'll tell you why. But anyway, so now you know how they work with these toxic organisms. Here's a pathogenic part of the Pfiesteria that we're talking about. This one, by the way, is a relative of Pfiesteria. They call it pseudo-Pfiesteria. And this is called pseudo-Pfiesteria Shumway, named after another woman scientist named Sandra Shumway. Okay? And that's another scanning electron micrograph. It looks slightly different than the other one. Now, here is something real unique. This shows you what happens in the form where it wants to gobble up something. This is a zoospore of Hysteria shumway. It's consuming red blood cells, okay? What happens is, now we're getting to be, sounds like science fiction here, okay? Notice what's happening. The zoospore, which are these little cells is extending something out, something like a trunk or something. And it's actually gobbling up the goodies inside the cell. This type of process has a word I'm sure nobody has ever heard of before called mycocytosis or vampirism. Okay? What happened here is the predatory cell, which in this case is the Pfiesteria, pierces the cell wall or cell membrane of the prey, which in this case is the red blood cell, and it's sucking out the goodies in there by a special structure called a conoid. And it sucks out the cellular content and digests it. You know, this would make a wonderful story for a science fiction movie called The Attack of Pfiesteria, the Cell from Hell, starring David F. Arieti as the professor. It may look something like that. Yeah. So, anyway, by the way, my middle name begins with F. You can take your pick. It either stands for Fabulous or Franklin. Either one. Anyway, so there's me in the lab, and there's one of the secretaries having to pay me a visit and being attacked there. Anyway, so, you know, you gotta be careful with this stuff. Okay. Now, by the way, if you're ever in North Carolina and you pass a crab house, just make sure you have this sign. No Hysteria here, lunch buffet. Hey, you know, I didn't make this stuff up. Right? All right. Now, here's some more things you got to worry about, as if I didn't give you enough. Here we have the erythromatous papules on legs of somebody, which means that this gives you little red spots if you go swimming into the water, okay? With this stuff, okay? You can get this. It's a real nice thing to have. Now, here comes parts of the human race which I really don't like. You know, I really get disgusted sometimes with the human race because the things we do. Okay, there's an organization called USAMRIID, which stands for the United States Army Institute, Marine Institute of Infectious Diseases. Okay? They actually dealt with the Ebola virus a number of years ago in the 80s. I gave a lecture here a few years ago about Ebola, and I actually showed the TV show based on this. It was a show called Day One, which was something like 20/20 or 60 Minutes. It was one of these type of expose shows, and they mentioned about that we were actually had an event where Ebola was brought to the United States in monkeys taken from the Philippines to do research on, and all the monkeys died because they had the airborne type of this Ebola virus, which was named the Reston strain because it took place in Reston, Virginia. And we were lucky that the human form of Ebola is not airborne. At least they think it's not. Anyway, so they mentioned a fellow named Peter Jarling who worked at USAMRIID, and he was actually the main character in this particular show. It wasn't a regular movie, it was a documentary, you know, and they were explaining, and he was explaining everything. Anyway, so that's when I first heard of USAMRIID. So, let me explain something. You all know we have the Pentagon, and they're finding new ways to kill people quicker and easier. One of them is aerosolization of bioweapons. A bioweapon is when they want to use some sort of biological substance. We call this weaponizing bacteria, weaponizing Ebola, weaponizing something, okay? And this form is aerolization. So, I want to show you what happened. This I got from the book, and the waters turned to blood. Now, this really made me angry, and it probably increased my blood pressure. Let me read it. Now comes the part that makes one wonder, is there intelligent life on Earth? How about weaponizing Pfiesteria? Okay, this is the part that got to me. I'm going to read this to you. Okay, this again had to deal with Joanne Burkholder. Like I say, she was the hero with this, and she had to put up with all this nonsense, you know? Let me read it to you. Dr. Burkholder went to a banquet after a conference about the oceans and pollution when a suavely handsome fellow, that's what they said in the book, dressed in casual sports clothes, walked up and told her how much he enjoyed her presentation. She wound up sitting at the same table as the man. He asked Dr. Burkholder, "Who's doing the toxin analysis?" She said a famous toxicologist named Dan Beatty would do it. Then he asked, "Why don't you let me do it?" Then she asked, "And who are you?" He said that he was a toxicologist at USAMRIID. There is the full name, United States Army Research Institute in Infectious Diseases in Fort Detrick, Maryland. She didn't know what went on in Fort Detrick, but what she heard made her uneasy. Makes me uneasy too. The Army worked with a lot of deadly toxins. They told her that they found a way to use toxins associated with the red tides for military purposes. And when civilian scientists interested in developing deterrents to red tides had requested information from the Army, they were told that it was classified. Then the man said, "Why don't you let Baden work on the toxin and let me handle the aerolization?" And so she asked, "Why are you interested in that part?" And he said, "If what you say about short-term memory loss is true, can you imagine its value to biological warfare? We could hit people with that and they wouldn't remember what happened." Now this to me, you know, it seems to me that we should be doing better things than this. You get the idea? I mean, than warfare. You know, I tell you, I'm very opinionated. And as I tell all my students and stuff, if I want their opinion, I'll give it to them. All right. Anyway, so here is the place USAMRIID, what it looks like. Now, here is a chart which shows you the 23 or 24 forms in various stages. It's quite interesting how they figured this out. But of course, the main creature at this time is this one there. That's that Lobos amoeba right there. See it? That one? And I'll go over here. Here's the Lobos amoeba there. See it? That's the main form which causes some problems. Quite interesting. Now, if you're interested in learning this, since Virginia is near North Carolina, they have this interest here, this little document here that I want to point out. And this tells you Virginia Department of Health, what you should know about Pfiesteria and Virginia water. This I just got up a couple days ago. So if you're interested, there's a lot of stuff here you can get. See, it tells you all about it. And by the way, PLO stands for Pfiesteria-like organisms, okay? And they say, those of you who like seafood, it tells you here, in general, it's safe. As always, you should not eat fish or sell fish taken from areas where large numbers of sick or dead fish are. So, you know, I don't know what to tell you. I myself don't eat that stuff. I don't eat clams or anything like that. Let's get back to this. There we go. And by the way, now, there's something interesting. In 1980s, I happened to be walking in Milwaukee. No, it wasn't the 80s, hold on. It was in let's see, I had my first book published in 2005. All right. It was around 2004, 2005 when I was writing my book. So, you know, I live here in Skokie and I hadn't been to Milwaukee a long time. So it's a true story. So I actually I took the train, Amtrak. Amtrak and Metra have got me well trained. Just thought I'd mention that. So I went up there. I was walking around Milwaukee because I wanted to see it. And lo and behold, I couldn't believe it. I ran across this hotel called the Hotel Pfister in Milwaukee. It's still there. And this reminded me of the organism Pfiesteria. Notice there's only one difference in the spelling, and that's the letter E. E is missing. So what I did was, I do stuff like this, see? It's missing. I went to the hotel. I said, I want to speak to the manager. I actually did this, and I told her, I think it was a woman. I don't remember, it was a while ago. She said, you know, I'm writing a book, and I'm mentioning this organism called Pfiesteria, and by coincidence, your hotel has almost the same name as that organism, you know, and she got a kick out of it. Yeah, I told her. I said, it's really a fancy hotel, but I don't think they have Pfiesteria there anyway, but, you know. Anyway, so I just told her that, and I thought I got a kick out of it. I think she got a kick out of it, too. I actually told her. Anyway, I do stuff like this. So, anyway, I think that's all I got to tell you. Now, if any questions, and by the way, I have my business cards here. I have plenty of them. So if anybody has any questions, comments, I have my books here. Go ahead and ask. Any questions? None? Yes?
How do you think we solve this?
What are we going to do about it? Ah, very good. I'm glad you asked that. First of all, we have to worry about pollution and climate change. That's the way to do it. Because I believe we are helping these things to evolve. You know what I'm saying? And I think the answer is to stop that. And by the way, I think that is the only answer. And I'm glad you asked that. Now, if we have time, I can show you the 20-minute show that I saw, and I recorded it a number of years ago, from this about Pfiesteria, if anyone's interested. You know, we can watch it here because I have it on Dropbox, the TV show. It's a 20-minute show from, it was Dateline was the show that I first got this from. And you'll see a lot of the people involved in that if you're interested. You interested in watching it? I'll put it on, then you can watch it.
Is the Chicago Lake or the river too?
It could be. I've never heard it here, but that doesn't mean it can't come here. So anyway, this is a real baddie, and I don't know what we can do. But you asked me, I think the answer is to clean up the water, you know? Because we're really messing around. We know what we're doing, but we got to stop it, you know? Okay, so I'll play this if you want. You could. Before I do that, are there any other questions, comments, complaints, anything? Yes, sir.
You mentioned the effect of all the hog farms.
Oh, yeah, I forgot about that.
And the hog industry. And
Yeah, farms here. Hold on. I did have pictures of hog farms. I'm glad you mentioned that. Hold on. Yeah, I forgot about that. He's right. You know, for some reason it disappeared. I put them on here. Maybe I put the wrong one on. Hold on. I did have hog farms, right. I'm glad you told me. Part of the problem, let me get this up here. I thought I put them on here. I guess I forgot to put it here. Yes, the problem is the hog farms where they have a lot of pigs and they raise pigs, and then you have a lot of rain, and what happens is the waste from the hog farm goes into the water. See, that's the problem. I'm glad you mentioned that. And the Pfiesteria love the waste of the pigs, you see? And what happened is, I had a good picture of that. Somehow I misplaced it. And I'll show you the hog farms when they had a flood, and then you can imagine what's going on. Because North Carolina is known for its hogs. And I did put that on. I'm glad you mentioned that, Pete, because I had it here. Let me get the hog farms, and I'll show
Did
you.
he kill the hogs?
Well, what's that?
Did that stuff kill the hogs?
Yeah, but they're not really located. The hog farms, they have the lagoons which hold the waste, and that breaks and goes into the river. So they themselves do not do that. I mean,
and the hurricane didn't help that.
yeah, right, exactly. That's the point. The hurricane, let me show you flooded hog farms.
Then what happens with the red tide? That goes away? That's a different thing.
Eventually. Now, I'm glad you asked that. What happens is, there are things which destroy the red tides. These are actually bacteria and viruses which actually destroy it. That happens automatically. That's a natural process that nature put in. Yeah. Yes, so you asked a good question, Valerie, and that's what happens here. Let me show you the pictures here. Hold on, I see flooded here. Let's take a look. These are the pictures I meant to show. Somehow, there it is. That's what I want. See that? Here are the hog farms. Look at that, folks. So, if you see those pictures, you can figure it out what's going to happen. You see it? So, all the waste goes into the Neuse River, and it's a gourmet restaurant. Okay. So, are there any other questions? All right. Well,
If
yes.
this has been proven, right, it's proven, but what is our government saying about it?
That's a good question. I don't know. We have all these problems with the government, especially now. I don't know if they want to clean it up or not. Oh, one of the issues is overpopulation. We have too many people. By the way, we have a bean problem. Do you know what that is?
Human being.
Human being. But anyway, and you know, they raise pigs because it's relatively inexpensive. We shouldn't raise pigs. I don't think pigs are healthy for people, okay? That's why it's so cheap, I think. you know, I myself don't eat them, but the point is, it's not healthy. But you can imagine after a rain, Valerie, see that? How that can go in there. And so anyway, now, do you all want to see that movie from the TV show? Yeah, I recorded it. Now, I admit the quality isn't that good. That was done over 20 years ago, and I had a video machine, which, you know, I didn't do a good job of it. So let's go to Dropbox. And I think you'd enjoy this. This is where I got a lot of the information, and then I delved more into it. I realized this is an issue that we should be worried about. All right, let's go to Dropbox. Sign in. Wait, I gotta go to a different one.
You just know.
Yeah, wait, I gotta go back up
No,
here.
I read the book.
Yeah, the book, Dropbox.
But the Tribune actually covered the yellow and white.
There it is. There it is. Sign in. And this you'll enjoy, I hope. There we go. Let me sign in. And we got the cap. Hold on. There we go. Let's see if that works. Oh, I don't save it. Is that working? Oh, is it working? There it is. Okay. Let's look at Pfiesteria. Let's see if that works. There it is. Let's see, do we have it? Oops, that's not it. That's the picture. I gotta find the movie. Where are we? Gotta find it. Here it is. That's it? Okay. All right, here it goes. Ready? And go.
On it about something terrifying in the waterways of North Carolina. I know. The same waterways that people use every year for boating, fishing, and swimming. Fish are dying, and now experts are wondering, could humans be at risk? And scientists who found some answers now have a question of their own. What are we prepared to do to fight back? The story now from Dennis Murphy.
See you guys. Thanks. Sorry for the quality, but
It seemed like there was something alive in my body. And that's exactly what I remember telling some of the doctors. I feel like this thing is alive.
Gail Jorgensen was sick for more than a year, and so were her 2 kids.
The children complained like electricity was going through their bodies.
And when they backtracked the mysterious illness that stumped doctors and specialists, they kept thinking about that river in North Carolina they put their hands in.
Gail, did the water look at all odd to you as you think back on it?
No, I mean, it looked like any other river, but then my children discovered dead fish.
Dead fish, fish with sores. North Carolina fishermen have seen them by the millions over the past 5 years. Fish kills that strike usually in the summertime. Marine scientists routinely blame low oxygen in the water. But Carolina fisherman Roy Rice thinks there's something in that water.
It acts like acid. It burns. The water burns you.
Something that's causing ugly, slow-healing red sores on his arms when he pulls at his nets. Red sores that have been showing up on fishermen and others who frequent the rivers and sounds of eastern North Carolina. Not everyone gets them. Just how many and just how often remains a mystery. But the symptoms are so troubling that last week more than 100 doctors from around North Carolina's river estuaries petitioned the state government to do something about it. Dr. Chris Delaney and his colleagues have treated a number of fishermen lately. The lesions that you're seeing, these red sores, the fish also are showing sores. Are they the same? Different?
I can only speak about my personal experience, and there was, visually at least, a remarkable similarity between what had happened to this fish and what was happening to my patient.
What's in the water here? That's a question that more and more Carolinians are asking themselves these days. Something more than the heavy metals and pollution that are poisoning so many of America's rivers. No, this is still a mystery. What's causing the fishermen to be sick? What's making some people complain about muscle spasms?
sorry for the
short-term
quality,
memory loss?
but best I can.
There is a person in the state who says she knows what it is. It's in the water, it's alive, and it's definitely a bad actor. She says she's even taken its mugshot.
It's a very tiny protozoan animal. Thousands of these little animals will fit on the head of a pin.
A one-celled marine animal in the red tide family called Hysteria, fish killer. Its weapon is poison. How can a little creature this big kill a fish this size?
Because the toxin is extremely potent.
So potent that when Dr. Joanne Burkholder, a marine botanist, studies living samples of Pfiesteria in her lab at North Carolina State University, she must wear biohazardous gear. This video shot in the North Carolina River shows what happens when Pfiesteria goes on the attack. The fish swim in a dazed circle. Within minutes, they're dead.
Joanne, this organism, your informed opinion, can it hurt human beings?
In my informed opinion, it can very definitely hurt human beings.
Can it maybe kill us?
It's possible, but there have been no deaths that we know of so far from it.
But she knows from first-hand experience it can make humans very sick. Early on in her studies, Burkholder and her research assistant were poisoned by airborne Pfiesteria toxin.
We developed sores, disorientation, memory loss. I was afraid to talk to people on a telephone because I realized I couldn't remember what they said at the beginning of their sentence by the time they got to the end of it.
And it was worse for her research assistant.
He basically collapsed. He could not remember how to find his way home. He wasn't breathing very well. He couldn't remember how to dial his telephone number. He lost the ability to read.
Both the scientists recovered. It took the assistant about 3 months. But both still suffer from lingering effects, muscle twitches, burning skin.
We do have enough information to know that we've got a real monster on the loose.
Rick Dove is a true believer in what Burkholder is finding in the lab. Dove, a retired Marine colonel with 2 tours in Vietnam, has seen Pfiesteria in its natural environment, the rivers of North Carolina. And he says it is fierce. It can bore right through a fish.
Puts a neurotoxin in the water, and literally, at first, I mean, when you're out on the water, you can see fish attempting to escape the water, literally jump out of the water or beach themselves.
Yeah, something behind the lab.
Well, it's like something's after them, and they know it.
Dove is paid to keep his eyes open for strange happenings in the water. He's the citizen watchdog on the North Carolina River called the Neuse.
You're the river keeper. How sick is your liver? She's really on the brink.
Rick Dove says he suffered from the same memory loss, disorientation, and red sores that Burkholder got in her lab. And he's talked to dozens of tourists and fishermen with the same symptoms, sometimes symptoms that last for years.
Would you tell your friends to come down to the boat ramp, put the boat in the water ski for today? No, I wouldn't. I do not use it. I do not put my hand in it. I do not eat the fish from it because I don't trust it.
Take that chopper ride up the Neuse, and it's not hard to see what's happened. Retirement and golf course communities have sprung up almost overnight. Their fertilizer runs into the river, along with municipal water. Add to that a new plague, corporate hog farms with open lagoons of animal waste, some of which can leak or spill into the river. And who loves all that pollution, all that nitrogen? You guessed it, the one-celled creature called Pfiesteria. Dove thinks they feast on the river's nutrients.
It has probably been around since the beginning of time, like the roach. But when you feed roaches, when you leave a lot of food around your house, what will happen? You have a lot of roaches. And I think that's what's happening here.
In her lab, Burkholder has confirmed just that. Pfiesteria she's found thrives on pollution nutrients. She gave me a demonstration of just how hungry these creatures are. She took a few drops of my blood, mixed it on a slide with some non-toxic Pfiesteria, and when I looked through the microscope, an all-you-can-eat buffet was underway. The spiraling blob is the Pfiesteria.
It's just absorbed on the white blood cells.
It just suctions the contents.
This is very odd. I'm watching this thing eat my blood.
All of which makes you wonder if this ravenous little creature is in the water here in North Carolina. Are we safe playing in the state's rivers? Are we safe eating the fish that come out of them? We asked the state's health director whether we knew enough about Hysteria to be afraid of it.
I think in the laboratory setting, it can hurt humans. I think it has hurt humans. In the environmental setting, I think the jury's out. I think it may hurt humans.
We asked Dr. Ron Levine to compare a photo of a sore on a fish with the photo of a sore on a fisherman. Could Pfiesteria have caused both?
To a layman's eye, that kind of sore, that kind of open wound, looks a lot like this kind of wound that you see on a crabber.
Yeah.
Can we make those connections? We can investigate to see whether there is a connection. Things may look very much the same, but not be related to one another.
While there have been no deaths, Levine says studies are now underway to determine just how Pfiesteria makes people sick and how many people have been affected. Until then, the state's Division of Tourism is referring all calls to Dr. Levine's department, and the only advice the state health director has is to avoid the fish kills. But Rick Dove says you can't always see fish kills when they're happening. He wants action.
Instead of the guys in Raleigh, the bureaucrats and the political leaders, instead of them saying, yes, you're right, you do have a problem, and we're going to fix it, they played the ostrich. They stuck their head in the sand.
Some government employees, speaking off the record, say they tried for years to get decision makers to take notice of Pfiesteria. But instead, they say the bureaucrats went after the messenger.
Were the officials interested in what you had to say?
I did not receive a very warm reception, no.
How cold was it?
It was fairly frigid.
And while Burkholder was battling with state officials, Pfiesteria was having a field day. In 1995, it was found that some of the worst fish kills in recent memory. Tens of millions of dead fish, and a lot of potential exposure to tourists like Gail Jorgensen, who had no idea what was making her sick.
My children were sick, and there was nothing I could do to help them, because I couldn't get answers myself.
Gail Jorgensen's story, along with a scientist and environmentalist battle with state officials over what to do about Pfiesteria, is the subject of a new book, And the Waters Turn to Blood. Author Rodney Barker says North Carolina officials ignored Pfiesteria, feared it would upset their billion-dollar seafood and tourism industries.
They did not conduct any responsible investigation because they were concerned about the implications if in fact they found there was this organism out there that's causing public health problems.
Levine, a health director, says that's not true.
To have a woman like Joanne Burkholder who finds something and says, this is alarming, we all together need to do something about it, and she says she gets beat up and is told to sit down and be quiet. I mean, how do you reconcile that? Well, she's never been told to sit down and be quiet by the public health system of North Carolina.
But Burkholder did receive a letter of apology from the state that seems to indicate that she was treated unfairly. The letter said in part, I'm sorry to find that some staff may have disparaged your work. I deeply regret those past problems. And Levine adds that while it took time to get money for research, Pfiesteria was not ignored. Since Burkholder first saw the organism swimming in her microscope in 1991, other scientists have verified her research. And now she's joined by a Marine commander, taking her information into battle with the state bureaucracy.
You don't strike me as a tree hugger. Not at all. You're probably not the kind of guy they're used to hearing from this kind of stuff. No, I'm not. I'm a Republican. And I'm a capitalist. And I believe that industry's good. And I believe that development's good. But it's got to be done right or we're going to shoot ourselves in the foot. You know what the guys in Raleigh are going to say? Rick Dove, you're killing us. You're going to murder our fishing business. The tourists are going to go right past the state with the boats on the trail. Then fix my river.
What does the future hold? Rick Dove believes this summer could be the worst yet. That's because Hurricane Fran and Tropical Storm Josephine overflowed a number of hog farms and waste facilities, dumping excess waste into the water that could trigger more Pfiesteria during the hot summer